we go. Yeah, should be good. Okay.
Thank you. Yeah, and if you guys have any questions, just pop them in the chat. Hopefully someone will answer them in the chat, or we'll answer them at the end of each topic.
I'll send a link.
Yeah. So we'll send the link of the slides to the
Yeah, Rahul, we're just putting them onto the drive, I think, right now.
Yeah, we need you, brother. It's your intro.
The intro.
Okay. So basically, right. So we are PSPiñata. So I'll take it away to Yajat, Mr. Dua. So
What am I doing?
introduce yourself, bro.
Okay, my name is Yajat.
Camera.
I'm also in second year med with these guys, unfortunately. Yeah, so what is it? Basically the dad, sure.
Yes.
Pretty much, yeah.
I moved here from Abu Dhabi last year. So I'm new to Melbourne. And yeah, moving on to Natasha.
Hi guys, I'm Natasha. I'm from Malaysia, so I'm currently in the process of swimming to Australia because there's your borders that aren't open and that don't want me. And yes, I'm very salty about it, but it's okay.
Wow.
Okay, moving on to Reesa.
Yeah, hi guys, I'm Reesa. Apparently I'm an extraordinaire. There was something else written there, but I didn't want that to be shared. And yeah, good luck. I mean, congrats for getting into med, and wish you all the best for this year.
Yeah, so I'm done. My little thing there is let's get smashed. Basically, may or may not be an alcoholic, but you guys will come to learn that pretty soon. But yeah, like, you know, congrats on getting into med, guys. You've done the hard yards in year 12. And you probably heard this from like everybody, but trust us. It's like really fun, and enjoy first year. Like, have the first year that we didn't have last year.
Yeah.
You know, if you can come onto campus, come onto campus. If you want to go to events, all that sort of stuff, because like it's really important. And yeah, make sure you get smashed. Oh wait, no, sorry, it's smash med shit. I'm not supposed to say that.
We're not promoting alcoholism here. My name is Pamali. We are, just like everyone said, congrats, you've done all the hard work, you're here now. You know, just my main piece of advice is, you know, work hard, play hard, just enjoy. When I say play hard, I mean like enjoy yourself. And you know, enjoy the first year of uni. It's going to be lots of fun. And if you need anything, we're all here for you.
Hi guys. Hopefully you're not sick of my voice already, but it's nice seeing you guys again. Basically Google, but don't take that too seriously. No, I'm joking. Yeah, enjoy your first PSP, guys. Enjoy your first week as well.
Awesome.
All right. Slides should be in the chat.
Yeah, so slides are in the chat. You can feel free to look at those. You don't really need to worry about notes too much. Just try and get used to the content, absorb it in. So today we're going to be covering cell biology, the cell cycle, HKS, which is
Health, knowledge, and society. Yeah.
And cell physiology. So starting off with cell anatomy. So Reesa, take it away.
Okay, yeah, I'm going to be taking you guys through cell anatomy. I just wanted to say I didn't do biology in high school, and it was fine. And the first year is also all pass fail, so I wouldn't stress too much, especially Sem 1 content doesn't really come up ever again. But I guess it is good to know the basics of what cells are and like foundational knowledge. Okay. Who's switching the slide? Okay, so first off, what is a cell? So there's two theories, but the one that I would recommend you to know is traditional cell theory. So 3 key points. All life is composed of one or more cells. Cells are the smallest unit to be considered living, so the basic building blocks of life. And all cells come from pre-existing cells via cell division. And the modern cell theory just kind of builds upon this to say that, you know, you have genetic material in there, cells kind of function similarly to one another, but the one I would focus on is traditional cell theory. So inside a cell, we have a couple of things. The first thing we have is cytosol, and this is like the watery fluid, the gel-like fluid inside of the cell, and it contains the organelles. So when I say organelles, I mean like these subcellular structures, and they each have their own specific function. And each of them are also membrane bound inside the cytosol. And we'll be going through a list of them that you guys will need to know later. Cytoplasm includes everything in the cell, including the cytosol, including the organelles, and the cytoskeleton, which we'll come back to later. It doesn't include the nucleus, however, because the nucleus is also separately membrane bound. And then the last thing in the cell are these like kind of random inclusions, so these inactive accumulations of like cell products, so like carbs, proteins, lipids, that kind of just in there, but that don't really do anything. So you have prokaryotes and eukaryotes. So the main difference that you need to know between these two is prokaryotes do not have a defined nucleus or organelles, and they're unicellular, whereas eukaryotes contain a nucleus and organelles. And that's the biggest difference. If you can just remember that, then you're set. But in that table, I've also made like a little summary of some other differences between them. It's also good to know what organisms come under each category. So under prokaryotes, we have bacteria and archaea. No, and bacteria, the second one's never come up before for us at least. And with eukaryotes, animals, plants, fungi, and protists. And this will come up again for you guys in microbiology. But yeah, differences, the nucleus, no nucleus, organelles, no organelles. Okay, so when we move on to the structure of a cell now, surrounding the cell, we have a membrane, and this is kind of like the skin. So just like our skin, it acts like a barrier of things coming in and out of the cell. And the reason it can do this is because it's arranged as a phospholipid bilayer. So I've included a picture of what a phospholipid looks like. So that blue part is the phosphate group, and it's hydrophilic. So it means that it's polar. And then the tail is hydrophobic. So with the cell membrane, they're attached. There's a double layer, which is the photo on the next slide, with the polar groups facing outwards and the tails facing inwards. So that means the outside is all hydrophilic and attracts polar molecules, but the inside of the membrane is hydrophobic. So that makes it selectively permeable. Polar molecules require assistance to get through, like transport proteins or protein channels, but small molecules like carbon dioxide and water, they can move straight across because the inside's hydrophobic in the environment. The inside is a hydrophobic environment. So the next slide shows a picture, so you can see what I was talking about with those little heads on top and then the tails in the middle, and it's a double layer. The cell membrane is also referred to as a fluid mosaic model. So basically it's made up of other things than the phospholipids. There's also proteins, carbs, and cholesterol. In your lectures, you'll go through a bit more detail as to exactly the functions of those things, but this is a really good summary. As long as you remember that proteins are important for communication and transport, carbohydrates important for cell-to-cell recognition, and cholesterol for rigidity, then you're set. It's really interesting, actually. So the reason, because of this fluidity, because the cell membrane is so dynamic, if you insert a needle into the cell membrane and then take it out, the membrane will kind of close over that fluidly. So yeah, that's what the model is describing. Okay, so these are the list of organelles that you guys will be going through. There's the ones that are listed in the lecture learning outcomes, but you might come across some others. So yeah, we'll start off with the nucleus. This is the control center of the cell. So because it contains our DNA. So basically it's the boss. And some cells have multiple nuclei, some cells have none. For example, red blood cells, they don't have any nuclei. So in the diagram, you can see some different parts to the nucleus. So the nuclear envelopes, that's the membrane I'm talking about. The nuclear pores, the communication little holes between the nucleus and the cytoplasm, so the contents of the cell outside the nucleus. And the nucleolus is the site of ribosome synthesis. So the difference between the nucleus and the nucleolus, because they're both kind of little ball shapes, the nucleolus doesn't have that membrane like the nucleus does. And it's really important for ribosome synthesis. So it contains a lot of rRNA, whereas the nucleus contains our DNA. This diagram also shows you chromatin, and we'll come back to this in our central dogma topic, but basically as a little idea of what it is basically DNA, but it's wrapped about tightly around these proteins. But you'll come back to that in a later topic. So the mitochondria, everyone's favorite, the powerhouse of the cell, basically where we get our energy from. It transfers energy that we take in into energy that the cell can actually use, which is called ATP. Again, you'll come back to this in metabolism topic, which is very a content heavy topic. Mitochondria has a double membrane. It's passed down to us from our mothers, so maternally, and it has its own DNA and ribosomes. Yeah, and you'll learn more about the different parts and how that energy is produced in central dogma topic. And also, if anyone has any questions, feel free to pop them in the chat as well, and we'll answer them. So ribosomes are technically not an organelle, but they kind of just fall under this list because they're another thing floating around in there. The reason they're not an organelle is because they don't have that membrane around them like all the other organelles do. They're really important for making proteins. So they're made up of RNA and protein, and they can be found just floating around, but they can also be found on rough endoplasmic reticulum, which we'll cover. Again, you'll come back to this in central dogma because making proteins is really important, part of translation. And it's made up of two subunits, the large subunit and the small subunit. And you can see in that photo, like a little precursor to what you'll be learning later about the mRNA, that being translated to amino acids and all that fun stuff. Okay, so the rough endoplasmic reticulum, again, protein synthesis. The reason that's its function is because it has those ribosomes embedded onto it. And this is a really nice picture. You can see that the rough endoplasmic reticulum comes directly off the nuclear membrane, and that's usually where it's found. Yeah, the smooth endoplasmic reticulum. So as you can see, it looks a little bit different to the rough. It's more like these tubules. It's really important for lipid synthesis. So just think a rough is protein and smooth is lipid. But it does have some other functions that are listed there, but that's the main one you need to know. And it's just smooth because there's no ribosomes on it. So yeah, those are the main differences. Smooth endoplasmic reticulum comes up again in muscle when we cover that in SEM 2. So you can see like a lot of this is just basic knowledge that you'll definitely build upon in later content. Okay, the Golgi apparatus. So this is like the post office of the cell. So all that stuff, all the other things are producing things like proteins, and then the Golgi apparatus receives it, kind of modifies it, processes it, where is it going, who needs it, and it just sends them out for release. So it's, yeah, like a post office because it's packaged at that cis face, so it's labeled in a diagram, and then it's released from the trans face when it needs to be. Yeah. So lysosomes, I kind of like these because they're like the recycling bit of the cell. They basically just break down useless stuff that no one needs anymore, and they break it down back into its original basic units, and then they're reused. So it's kind of an efficient system. And it's the way it does this, it's filled with enzymes called hydrolytic enzymes that do this. Lyse, L-Y-S-E, that means to break down, that prefix. So if you ever see lyse, it just means breaking down. So lysosomes break down products. And finally, secretory vesicles. So these are found in cells when we need to store something for a while before it's released. So you'll come back to this when we do neurophys, because our hormones, our neurotransmitters, they're stored in these. But they're also produced by the Golgi apparatus. And you can see they're these little bundles, and then when they need to be released, they go to the edge of the membrane and kind of dock there, and then the products are released. So this is a summary I made last year because I didn't do bio, so this was all like new for me. So I'll be putting on the Google Drive after, just like a little picture with all the little organelles you need to know. Yeah. Okay, the cytoskeleton. So like the name, it's just like, it's like the framework, the bones of the cell. It's really important for the shape, the structure, and also cell movement. So the three things that make it up are microtubules, intermediate filaments, and microfilaments. So the ones I would say, the most important ones out of these that you would probably need to remember are microtubules and microfilaments, because microtubules come up in mitosis, and microfilaments will come up in SEM 2 again when you're studying muscle. And yeah, so it goes from largest to smallest, and you can see what they kind of look like, hollow tubes versus intertwined strands. So microtubules are important for maintaining cell shape. but also really important for cell motility and, as I said, mitosis. So during mitosis, they aid in dividing the cell by helping separate the chromosomes when the cell divides into two, which we will go into a bit later in the PowerPoint when we cover mitosis. So basically, a centriole is just 9 microtubule triplets in like a little circle or pinwheel. Yeah, and that's basically it. Yeah, these two things are really important. So cilia, they're also made from microtubules. They are like these little hair-like protrusions from the cell surface. And they can either be motile, which means that they help move things. So for example, in our airways, when we get dust or particles in there, they help move them back out so that we don't get like infections and things like that. Or it can be involved in cellular signaling. So this is when they don't help move things, but they're important for sensory information. So for example, there's some cilia in our kidneys, which helps signal when there's like a flow of urine and stuff like that. The flagella also are important for movement. So on sperm, at the end, you'll have like a little tail, as you can see, but also in bacteria, it helps to move around. Both of these are arranged the same way, which is helpful to remember. Just remember as a 9+2 arrangement. So 9 pairs in the circle and 1 pair in the middle. Yeah. That's it. And microfilaments. So these include actin and myosin. So this makes up the muscle cell as well. So you'll come back to this in semester 2. Actin is really abundant. It has a high concentration at the edges of the cells and really important for cell motility. Myosin is pretty not as abundant, but except in muscle cells, but not in all of other cells like actin. Yeah, that's it. Oh yeah, this is a summary of the different types of things making up the cytoskeleton if you wanted to know more about it.
Okay guys, so I'll be taking you for the cell cycle. So those of you who've done bio, maybe in year 11, may have done this before. For the others, it may be a bit confusing, but I recommend just trying to absorb it in for now and then come back later, read over the slides, use your textbooks and try and understand it. But hopefully today, if you guys can leave with a rough understanding of what happens to a cell in its lifetime, that's going to be good. Okay, so today we'll be looking at the 2 types of cell proliferation. So like Reesa explained in our cell theory, all cells come from pre-existing cells via this process called cell division. So we've got 2 types of proliferation. We have mitosis, which is for somatic cells. So these are like cells throughout our body, in our tissues, in our bones. And then we have meiosis, which is for to produce gametes, which go on to, make offspring. And these are generally going to be a bit different to the parent cells, while mitosis is going to be identical. So we'll start off with looking at the cell cycle. So if you guys look at that diagram, you'll see that entire gray area. So we have G1, S, and G2. This is what we call interphase. So interphase is the period between cell kind of splitting, and this is where we have these 3 phases. So in G1, the cells have to increase in size and prepare for S phase, which is where DNA undergoes replication. And G2 is really important because this is where we check for errors. We really don't want our cells splitting and proliferating while they're being errors. So this is what can lead to cancer and things like that. And you'll cover all this DNA replication later when we do the central dogma. Okay, so we'll have a look at the stages of mitosis. So to start off, we have the first phase. It's called prophase. So what happens here is that our chromosomes become condensed, and they form this double-stranded structure. And our nuclear membrane breaks down, and Reesa explained centrioles. So these go to opposite ends of the cell, and they leave behind these trails. They're called spindle fibers. And these spindle fibers are really important for our next stage, which is metaphase. So basically what happens here is that those fibers, they go and they bind at the center of these chromosomes, and our chromosomes beautifully line up along the center of the cell. So if you guys, for those of you who did bio, you guys might be familiar with this concept called a karyotype. So this is just like a snapshot of an individual's chromosomes, and it's really important for diagnosing any chromosomal disorders. For example, we have Down syndrome, which is one we look at later on in genetics. So after that, we have anaphase. So in anaphase, if you guys remember, so we have the spindle fibers that come from opposite ends of the cell. So what happens in anaphase is that the spindle fibers begin to contract, and as they contract, the chromosomes, they split, and like a leg from each goes to opposite ends of the cell, and at each end you have identical sets of DNA, and this is in preparation for a last stage of mitosis called telophase, where all our DNA is now replicated. It's at each end of the cell, and we get a little cleft forming, and we're prepared for the cell to now split into two. So this process of splitting into two, we call cytokinesis. And this is when the final step of like the cell cycle is finished and our cell is now two. And this can continue happening until until we have a sorry.
Jesus.
Anyway, this can continue happening until we have an exponential increase in cells in our body. Okay, and this is a really good acronym to use, PMAT, to memorize and remember the order. So we have prophase, metaphase, anaphase, telophase. That's PMAT. And there's a little diagram there just to, have a look at. Okay, so then we have meiosis. So meiosis, like I briefly explained before, it's very similar to mitosis, except instead of making two identical cells, we're making four. And this is for sexual reproduction. And instead of one cycle of PMAT happening, we actually get two. So we get two cycles. We call them meiosis 1 and meiosis 2. So meiosis 1, so in each step of meiosis, in each stage, we have PMAT happening. So in meiosis 1, we have, of course, prophase 1, metaphase 1, anaphase 1, and telophase 1. So it's very similar to mitosis. So if you guys memorize PMAT from mitosis, you should be able to understand meiosis very well. Just the main thing to remember that's different is that in prophase 1, we have this process called crossing over. And this is how our DNA gets variation. So this is why your siblings look nothing like you, even though you share the exact same DNA from your mom and your dad. So, come back and have a look at this. I won't go into too much detail because we basically covered it in mitosis. In meiosis 2, this is also very similar to meiosis 1. I'd say meiosis 2 is basically mitosis because there's no crossing over that occurs. It's just the cell splitting into two. If you look down here, you'd see that you start off with what we call a diploid cell. So you have two sets of DNA, and all your granddaughter cells, the four that you're left with, they're haploid. So they're left with one strand. So this means that they can combine with, so if this, let's say this is the sperm, it can combine with an egg and produce an offspring who has two sets of each chromosome. So let's have a look at genetic variation. So this is the way in which each human on Earth has their own unique genetic combination. So this comes from crossing over of chromosomes, and we also have independent disjoining or separation during meiosis. And this is quite important for later on as well. So non-disjunction, this is pretty high yield because it comes up later when you're doing genetics. So like if you guys remember, we just went over meiosis 1 and meiosis 2. So in each step, we have anaphase 1 and anaphase 2. So this process of non-disjunction is when there's a failure of those chromosomes or those sister chromatids to actually separate. So this means that there may be a cell, there may be a granddaughter cell that is missing a chromosome, or there could be one with an extra chromosome. So we have some disorders listed below. You'll cover these again when you go through genetics. You guys, I feel like most of you guys will be familiar with Down syndrome. So that's when you have 3 of chromosome 21, and we call that trisomy. And we also have Edwards syndrome, which is 3 of chromosome 18, and Patel syndrome, which is 3 of chromosome 13.
Right, Pamali, teach them the PEDOS acronym.
Oh god.
Oh god. So this is an acronym me and Reesa came up with. It's kind of stupid, but it really helped us remember it.
That ain't it.
Yeah,
Mm.
we just told
Oh,
everyone
no,
sends to
No, I'm pretty sure I got from another PSP.
another PSP. Okay, well,
Say it. Say
Okay,
the pedos.
Patel, it sounds like pedo, and a 13-year-old is
No, PEDO. PED.
was that it?
Yeah.
Yeah.
Pedo, Edwards.
Yeah.
Oh my god, I was using it that way. We
The
second year.
pedos like them young. So P is the youngest, so number 13, E 18, D 21. So it goes up. That's how you can
Oh,
remember it.
okay,
It stuck with me. That's the only reason I remember this.
I don't know. I was thinking it that way. That's my bad then.
I'm gonna move away. I'm
Okay.
so shame myself.
Hey, shut up. Okay.
Guys, there's so many weird acronyms in med.
That's just the way you remember them. The weirder it is, the most the better you'll remember it, honestly. Okay, and then we have our sex chromosomes, which are X and our Y chromosomes. So we can have 2 main syndromes from that. So we have our Turner syndrome, which is monosomy. So instead of having 2 X chromosomes, generally a girl is born with 1. And in Klinefelter's, there's non-disjunction, which leads to a trisomy. So you get XXY, and these are all covered in a lot more detail later. Okay, so next we have checkpoints. So these are just there to make sure that everything's going according to plan. Because like I mentioned before, we don't want cells that have, problems going on to proliferate, and we really don't want, tumors and chunks of our body being just stuffed up, basically. So there's two pathways. So if there's an error, we can try and correct it the best we can. And if correction is not possible, we'll undergo cell death, which we call apoptosis, which we'll cover soon. So this is very low yield. It was mentioned in a lecture once, never saw it again. So if you guys don't understand this, I barely understand it, so don't worry about it. But basically, cyclins, they're these proteins that help drive the events of the cell cycle. And we can actually have our inhibitors coming in and inhibiting the cyclin complexes, which will actually end up stopping the cell cycle. So that's how we regulate it. Don't worry too much about it, guys. It probably won't come up again. And then we have our inhibitors. So we have our B, which is active at the G1S checkpoint, and we have P53, which is actually a tumor suppressor, and that's quite important, and you'll come across that when you do cancer later on. Okay, so we also have oogenesis and spermatogenesis. So this is just the formation of eggs and sperm. This is also pretty low yield. I barely understand it again. So if you guys don't really understand, that's okay. I'd say remember the definition. So oogenesis is for eggs and spermatogenesis is for sperm. And yeah, so they happen at different times in life. So just have a look at this table later. But the main thing is at the end of oogenesis, you're left with 1 egg and 3 polar bodies. So only 1 egg carries everything needed for an embryo. And at the end of spermatogenesis, you're left with 4 sperms. So that's just the main takeaway from that. Okay, so this is cell death apoptosis. So this is what we call programmed cell death. So this is what we want this happening in our cells. We want, if there's any problems going on, we want our body to recognize it and be like, stop, we need this cell to undergo cell death. So that it doesn't replicate and cause disasters within our body. So we've got 2 pathways. We've got an intrinsic pathway, so which comes from within the cell, and that's due to like DNA damage. And then we have an eccentric pathway. So this can be from hormones, toxins, radiation, infection. So the first step is that we receive that signal. The second step is that the cell is going to shrink, it's going to lose contact, and our DNA is going to begin to degrade. What happens after that is that the nuclear membrane will also begin to degrade, and our cell is going to lose its volume, and everything is going to start clumping up. And step 4, this is what we call blebbing. So as the plasma membrane begins to degrade, it's going to form these things called blebs, which are basically like lumps and awkward bits of the plasma membrane. It's going to lose its integrity. That's the main thing. And after blebbing, our nucleus is going to collapse, and it's just going to start fragmenting, and our DNA is going to start fragmenting. And lastly, so when excessive blebbing happens, our cell is actually going to break apart into small little bodies called apoptotic bodies, and they're going to be engulfed by phagocytes. So for those of you who haven't heard what phagocytes are, they're basically these cells in our body that go around, they engulf material. It could be dead cells, it could be like even bacteria and things like that. They're just any cell that can engulf. And we have special cells that will engulf these apoptotic bodies, and they will break them down, they will digest them, and nothing's going to happen to our body. It's just a totally natural part of life. And yeah, so there's not going to be any inflammation, and it's all going to be good. But we also have a process called necrosis. So this is kind of the opposite of apoptosis. So like I said before, so apoptosis is very natural. It's going to happen. It's very controlled. There are signals to it. But necrosis, on the other hand, it's totally uncontrolled. This is due to pressure. It could be due to injury, infection, poison. It's going to lead to pain, and it's going to lead to inflammation. So basically what happens is there's going to be an injury to our cell. And our cell's first reaction to this, it's going to try and recover the best it can. And if it can recover, that's all good. It will go back to being a functioning cell. But if it can't, and there's a lot of bad damage, what's going to happen is that the cell is going to swell, and eventually it's going to burst. And when the cell bursts, it's going to release its intracellular contents, and this will generally cause inflammation and damage to even our surrounding cells. And that's it from me. So I'm going to hand it over to Natasha for physiology.
Okay, guys, I'm going to take you through body fluids, which is like a basic physiology concept, which you'll hopefully use in your later years. Yeah. Can you change the slide? Yeah, so about 60% of your body weight is water, and of that 60%, one third is extracellular fluid, which then is split up into plasma and interstitial fluid, and then 2/3 would be intracellular. Osmolarity and osmolality are basically the same thing, but in different units. So osmolarity would be in terms of volume, and osmolality would be in terms of mass. And it's important to remember that osmolarity of red blood cells is 300 milliosmoles per liter, because you'll use that in further calculations. Effective osmoles is basically solutes that don't move across a semi-permeable membrane. So you know how Reesa mentioned that it's a phospholipid bilayer. So things that are hydrophobic generally don't move across. So these would be your effective osmoles. And a substance is effectively impermeable when it can diffuse across a membrane, but it's pumped back out. So a good example of this is the sodium-potassium pump. So sodium is pumped into the cell, but it's pumped back out. And potassium is pumped out of the cell, but it's pumped back in by the cell membrane itself. Yeah. So there are a couple of ways that substances can move across cell membrane. The most obvious one is diffusion, which is down a concentration gradient. So it's a passive movement and it doesn't require energy. And There are a couple of factors that you need to remember. Honestly, the most important one is temperature and the steepness of the gradient. So if there's a bigger difference in concentrations, then diffusion would occur at a higher rate. Hydrophobicity is something.
This is like a peer support program, so it's like year 2's teaching us.
Yeah. So hydrophobicity. So if the substance is hydrophilic, it would move. It would move pretty fast. But if it's hydrophobic, it wouldn't. Yep. So osmosis is movement of water molecules from a region of high from a region of low solute concentration to high, and this occurs through a membrane. I'm sure you've heard of this before. Facilitated diffusion is the movement of see those substances that are either too big or charged or polar or hydrophilic. Those substances would need a membrane protein such as a carrier or channel protein to enter the cell. And then active transport is an active process, as the name suggests. It requires energy in the form of ATP, and it moves against the concentration gradient. Yep. So osmotic pressure is a very low yield concept, honestly, but it's the pressure due to different concentrations of non-diffusibles. And basically it's what you need to apply to the solution to stop osmosis from occurring. And yep. Tonicity is basically the osmolarity of only the non-diffusibles, and it has two solutions would have the same tonicity if there's no water movement in between them. And yep, the tonicity of red blood cells is also 300 milliosmoles per liter because all the osmoles are non-diffusible. And yep. So this picture you'll come across quite often, I think. So if a cell is in a hypertonic solution, that means there's more solutes inside. Wait, sorry, give me a second. There's more solutes in the solution rather than in the cell. So there's higher water potential in the cell, meaning that water would move outwards. And in an isotonic solution, there's no net water movement. And in a hypotonic solution, that's basically the opposite of the hypertonic, where there's more solutes inside the cell. So more water would want to come in to sort of reach equilibrium. And it's important to keep in mind whether they're referring to the solution or the cell when they say hypertonic or hypotonic. Yep. So for a red blood cell placed in pure water, the initial osmolarities of the red blood cell, as I mentioned earlier, is 300, whereas water would be 0 because there's no diffusibles or substances in it. So finally, it would be 300 and 0 as well. So water would move into the cell and cause a lysis. Water is both hypoosmolar and hypotonic. So yeah. Next, when you put a cell in saline, saline is isoosmolar, so it has also a osmolarity of 300. And sodium is effectively impermeable, which means that, like I mentioned earlier, it is pumped in, but it's pumped back out by the cell membrane. And if sodium doesn't move, then the chloride ions in the solution also wouldn't move to maintain electron neutrality. So the final osmolarities would be the same as well. So there's no net movement, which makes it an isotonic solution. For a red blood cell placed in urea, 1.8% means that it's isoosmolar as well. So it has an osmolarity of 300. But in this case, urea is freely diffusible. So it would diffuse into the cell down its concentration gradient to reach equilibrium. And this means that the final osmolarity intracellularly would be 300 initially, plus 300 from the urea, so 600. So now that the osmolarity inside is higher than that of the outside, the water would move into the cell, making it hypotonic. Yeah, and this table basically explains what I just said, but it includes dextrose. So dextrose is a common intravenous solution that we infuse Dextrose is basically glucose, so it's used for respiration. So it's freely permeable, but it requires a transport protein. So dextrose, when it's used for respiration, the concentration outside the cells decreases. That means the water would enter the cells by osmosis down the concentration gradient. So this would cause an increase in total body water, meaning in ECF and ICF. So yeah, whereas saline, it basically, it doesn't enter the cells because it's effectively impermeable. So it would only increase the extracellular fluid. Yep. Yeah, this is probably the most important part. Isoosmolar saline is given to patients when they experience blood loss. And this is because it increases the extracellular fluid, and it's quite a fast process. So if people would have, if patients would have like a very detrimental wound, or like, for example, a gunshot wound, you would administer saline because it would increase the blood volume pretty quick to ensure that the patients don't go into shock. But whereas dextrose is a very gradual process because the dextrose needs to enter the cells via active transport and be respired before water can enter. So this is most commonly given to dehydrated patients. You can give saline to dehydrated patients, but it's just not that commonly seen. Yeah. You wouldn't give water and urea to patients because basically all their cells will lyse and you don't really want your patients to explode. So yeah. Yep, and now I'll take you through some of HKS, which is Health, Knowledge, and Society. And honestly, this stuff is really low yield. Like, you don't really need it in the grand scheme of things, but we'll go through it anyway. So according to WHO, which is the World Health Organization, health is the state of complete physical, mental, and social well-being, not just the absence of disease. But this definition has some flaws because, well, the concept of well-being may be different to everyone. And it doesn't really take into consideration like chronic diseases and things like that, because those affect your health essentially, but they're not like immediate and they don't, yeah. So equality versus equity. With equality, everyone is treated equally and it's assumed that everyone will benefit from the same supports or same resource allocation. And with equity, it recognizes that different individuals may require different opportunities because of their circumstances. So resource allocation would go in line with that, and there's distributive justice. So health inequities can occur at many levels. It can occur at a global scale, national scale, or even a local scale. And it can be avoidable or unavoidable. Like, avoidable differences may be like income distribution between genders, but unavoidable health differences may be things like genetic predisposition to some diseases. And yep, health indicators. DALYs is something that you might come across. It quantifies the burden of disease by giving it like a number, and it takes into account morbidity and mortality rates. And then mortality rates, this may be adult, neonatal, infant mortality rates. It's just, yeah, like a count of mortality basically. And then things like life expectancy, prevalence of disease, GP services, and frequency of health risk factors and comorbidities. These are indicators of how healthy a country or a social group may be. And then global forces affecting health equity. Can you change the slide, please? Sorry. Yeah, so these impact health systems, which then impact health outcomes. So geopolitics, these may include things like conflict, which may result in things like displacement and asylum seeking, which you'll cover in future weeks. And then sociocultural factors, which include like neglecting human rights and the social determinants of health, which we'll also go on deeper into. Environmental change, this affects the environment, which then also affects your health, obviously. Historical influence, this may include things like colonization and violence and stuff like that, which could affect your health. Yep. There are problems with global health, as we've seen in the like recent pandemic, is that most improvements have been centered on management of single diseases rather than improving the healthcare systems themselves. So this would result in too few staff, overworking, not enough infrastructure, and with things like clinics and PPE shortages. And when new diseases or pandemics arise, there would be like a spike in morbidity and mortality, which we've clearly experienced and is not fun. Yeah. That is basically it. Do you guys have any questions?
Yeah, guys, just ask anything if you're confused. Doesn't have to even be about the content. If you're confused about like uni so far, about like it's anything that you've
Like any questions,
Yeah,
any questions. in
literally
general.
anything.
we're here to
Or
help.
if you want to private message them instead, that's okay.
Yeah,
Yeah.
up to you.
Yeah.
We can all be a Google today.
Yeah.
Yeah.
Yeah, literally.
Any advice?
And then there was a lot of information, but honestly, like you're gonna go through it in so much more depth and like slower this week. So this is just like a crash course.
Yeah.
So a piece of advice from me is, watch the lectures. Sometimes when they pile up,
You sure?
Don't watch HEP.
Don't watch
yeah,
HEP. Don't
there
watch
are some lectures that are like really low yield. So
Like-
we'll tell you like what
Yeah.
subjects are very high yield. So from today's PSP, the cell stuff that Reesa covered and the cell cycle are somewhat high yield, and so was some. Do you remember that table that Natasha had with all the different solutions? So that's also high yield. But there's like HKS is a very small part of
Can you
your.
explain what do you guys know what high yield, low yield means? Or is that just random to you guys?
This should-
Yeah,
it's
it's
very
good.
important
I didn't know.
versus
That's
not
why
important
I didn't know
versus-
for like a good couple
Yeah.
weeks.
Basically,
I was just like,
what's
what
going
is
to show
this?
up on exams
Yeah,
versus
yeah.
what isn't. So as the
So-
year goes on, we will be putting
Mhm.
out hopefully question banks,
Yeah.
weekly question. Have we done any this week? I don't
No, we
think so.
No, we haven't.
So as on each weekly slide and each topic, we'll have a bunch of questions. So that should be a really good study resource. Testing yourself, if you find stuff online, these are very common topics across the world. So if you literally look up cell anatomy, cell physiology questions, go through them, see what you can do. If something is completely out of the blue, you probably don't need to know it. And
Yeah,
if there
that's-
is, just feel free to send it
It's like
over to us.
discussions on your group chat.
Yeah, and if you
Yeah.
guys are confused about like, oh, is this important? Because I remember,
Oh,
Yeah.
sorry.
because I remember last year like learning stuff, and I was like confused whether I actually need to know this for the exam. So just shoot one of us a message, just be like, hey, is this going to be important for Sem 1 exam? We can just tell you it's a pretty low yield thing, so don't worry too much about
And
it.
We'll probably set up like a curious cat thing. I think
Yeah.
that's going to set that up.
Yeah.
So that it'll be posted on our
It's basically an
group page.
anonymous Q&A. So
Yeah.
Yeah.
just go for it. Easy.
And like today's content, it's a lot like for this
Yeah.
week, right? But the most important thing I think right now is just take it slow. Don't try and learn everything off by heart. Just try and
Yeah.
understand as much as you can.
Yeah.
Like have
And
fun.
take it as it comes.
Yeah, remember like you have SWOT vac before your exams. You'll have
You have a mid-sem break.
You
as
have a
well.
mid-sem-
For it also all pass fail. Like
yeah, that's like the
Facts.
most
Yeah.
important thing.
Like
Yeah,
your,
I
like
mean,
your
you
grades
get-
don't really
it's-
count.
It's a
at all.
thing you'll keep.
because I would focus on just going to events and socializing, like do all that
Smash.
over studying.
I'm joking, I'm joking.
Um,
Have fun.
That was
Have
that
fun.
was my thought.
But
with that.
you know,
Nah, smash
study
that.
groups are also a great option. So when you guys get to come onto campus soon, hopefully, form some study groups with some friends. So that's a great way to, socialize, also
Yeah.
study, and teach your friends. That's also a really good
Mhm.
way. What else? Making
Yeah.
notes. Do what works for you. So if
Yeah.
someone's doing comprehensive notes of every single lecture, let
No,
them do that.
yeah,
Like let
yeah.
them do that, right?
It's not target,
If
is it?
Figure out what
It's not
figure out what
target
works
at
for
all.
you. Basically.
figure out what
Yeah.
works for
And you
you.
will. I mean, no
Yeah.
stress.
Yeah.
Basically, you'll keep hearing throughout this year, P's get
Reesa.
degrees. So as long as you pass, you'll get the degree.
Yeah.
No stress. I think what it's still
Like-
50% this year.
Yeah, it's still
around
50%, yeah.
50%.
So you just have to pass like literally the rest of it. What?
Yeah.
Yeah. But that's a long time away. That's
Yeah.
a long time
Okay,
away.
okay,
So yeah,
okay,
you guys should go
sure.
enjoy
Alright.
your first
Yeah.
week.
Anyways, yeah.
Enjoy. Reach out, you need anything. Yep.
No worries.
Any
Just a
any
Oh, he said
oh,
thank you.
oh, okay.
Yeah, all good.
All
All
good.
good.
All
Bye,
good.
guys.